Today we are going to study the next class of the plant growth regulators or phytohormones, that is cytokinins. Actually the word cytokinins is derived from cytokinesis. Cyto means cytoplasm and kinesis means divisions. Here cytoplasmic divisions. And these are the phytohormones that promote cell division in plant roots and shoots. So coming to the discovery of the cytokinins. First of all, it was Couch and Miller who observed the internodal region of stem of tobacco plant showed proliferation only if auxins were accompanied with some other substances. What Couch and Miller observed? They were observing the internodal region of stem of tobacco plant, and they observed that the proliferation of the cells occurred only if auxins were accompanied with some other substances, and what they did, they gave the plant the auxins plus the extract of the vascular tissue or coconut milk. That substance I'm telling you can be extract of the vascular tissue, coconut milk, or yeast extract, or DNA extract. Fine. So auxins plus that substance, and that substance can come from any of these tissues. And later Couch and Miller in 1955 isolated the substance from the herring sperm DNA. Herring is a fish and called it as kinetin. They called it as kinetin because of its ability to promote the cytokinesis, because of its ability to promote cell division. And this substance which they extracted from the herring sperm was found as 6-furfuryl aminopurine, and they called it as kinetin. And next, later in 1961, Miller and Latham independently found that the naturally occurring kinetin in plants and called it as zeatin. Fine, they called it as zeatin. They found it occurred in the zeamase. Zeamase is corn, and as the botanical name is zeamase, they named it as zeatin. So here is the structure of the zeatin. It's the trans zeatin and it is the cis zeatin. They are the isomers of each other. Fine. So this is about the discovery. Now coming to the structure of the cytokinins. Cytokinins are the adenine derivatives. You must have heard adenine is a purine base, nitrogenous base present in the DNA. Fine. So here is the adenine, and cytokinins are the derivatives of this purine base. Here for this I have taken kinetin, which was extracted from the herring sperm, and this is 6-furfuryl aminopurine. Here is the adenine. Fine. Here this NH2 group, it contains an extra ring. Fine. So it becomes 6-furfuryl aminopurine. So these are the derivatives of the adenine, which is a purine base. Now coming to the physiological role of cytokinins. Let me tell you, cytokinins, as I already told you, they promote the cell division. Fine. And they help in the formation, maintenance and growth of the shoot apical meristem. Cytokinins are the positive regulators of the cell division in the shoot apical meristem, and negative regulators means they don't enhance the cell division in the root apical, but they enhance the or promote the cell division in the shoot apical meristem. Now the next role is morphogenesis. Morphogenesis means shoot and root initiation in the callus. Callus is formed during the tissue culture from a single cell. A mass of undifferentiated cells is called a callus. So mass of undifferentiated cells is called callus. So the callus is a mass of undifferentiated cells. So when shoot and root initiation starts in this callus, this is called as morphogenesis. So when auxin is present or the auxin to cytokinin ratio is high, the callus will initiate root formation. Here you can say when the auxin concentration is higher than the cytokinin concentration in the callus, root initiation will start or the root formation will start in the callus. So this is the one condition. Second is when cytokinin concentration is high to auxin, means cytokinins are present more than that of the auxins, it initiates shoot formation. Fine. So this is the second condition. And number third is when auxin and cytokinin concentration is equal in the callus, there is no differentiation of shoot or root, only it promotes the proliferation of the undifferentiated cells of the callus. I hope this is clear. So moving next to next role of the cytokinins, that is the apical dominance. I have already told you that the apical dominance is when the apical bud suppresses the growth of the lateral buds, because the axillary bud or the apical bud contains the auxins, and that is responsible for the apical dominance. But here direct application of the cytokinins to the axillary buds inhibits the apical dominance. If cytokinins are applied to the axillary buds, that inhibits the apical dominance. As a result, what happens? Growth of the lateral buds occurs in many species. So, it is an inhibitor. Cytokinins, you can say they are an inhibitor of the apical dominance. Now, next we have chloroplast maturation. Cytokinins promote the conversion of ketoplasts into chloroplasts via stimulation of chlorophyll synthesis. You know, the seedlings which are grown in the dark, away from sunlight, you must have seen they turn pale. They elongate and turn pale. They don't they and turn pale in the sense they are yellowish in color, because they don't have the chlorophyll present in them, and the chloroplasts in those dark grown seedlings become the etioplasts. They become etioplasts because they lack the pigment chloroplast. And when these etioplasts or these dark grown seedlings are applied or given a dose of cytokinins, chlorophyll initiation in these plants takes place and the etioplasts become chloroplasts. Fine. So this is the next role of the cytokinins, chloroplast maturation. And next is senescence. They delay the leaf senescence. Senescence is aging process in the plants. So if we have seen, if externally a leaf is given the dose of the cytokinin, it remains longer, it remains green for a longer time, and as a result the process of aging is delayed. On the other hand, if the leaf is left as such, it starts to turn yellow, and as a result the process of aging or senescence takes place. So, what is the role of cytokinins here? They delay the leaf senescence. So, this is all about the cytokinins. If there is anything, please let me know in the comments. Thank you.